Good evening, my dear students. You are all warm welcome to the another session of the biotechnology and biology. Today, what we are going to discuss, biotechnology, the continuity. Last class, what we have discussed, what is meant by biotechnology, who gave you names, biotechnology. And the name of the scientist called us, what is that? Carl Ehrlich. He's a Hungarian engineer. In 1919, the term biotechnology, the word was given by Ehrlich. Okay, so that things already we discussed. And then the European Federation of the Biotechnology, EFP, has defined biotechnology as they have given as a definition of the biotechnology. What is that meaning? The integration of the natural science and organisms, cells, parts thereof, and molecular analogs for product and services. This explanation given by the EFB. Okay. And then, so next we will pass on to the continuity, particularly. So what are the types of biotechnology? See that the science of the biotechnology can be broken down into sub-disciplined. What are the sub-discipline is, So one is called as red biotechnology and white biotechnology, green biotechnology, and the last is called as blue biotechnology. Each one, it has a specific applications. Okay, so for example, see one by one, then only you may understand the concept. Why it is called as red biotechnology, why it is called as white biotechnology, why it is called as the green biotechnology, and also others. Okay, see that red biotechnology involves the maximum in the medical process. Okay, medical process, such as using organisms, to produce a new drugs, or using the stem cells to regenerate damaged human tissues. This is very, very important. Damaged. Okay, damaged human tissues. This is called as, what is that? This type of application is called as, what is that? Red biotechnology. Once again, repeat for you. Red biotechnology is involved in the medical process, such as using organisms to produce a new drugs. This is very, very important. Red is denoted as new drugs. Or using stem cells to regenerate damaged human tissues also. This is called as, what is that? Red biotechnology. Next one is called as white biotechnology. It is also called as, what is that? Gray biotechnology. What is the meaning of gray biotechnology? Where it is used or involved, what is the main application of this white biotechnology? See that once it involves the industrials. So industrials process such as the production of new chemicals or the development of the new fuels or for vehicles. So these are called as white biotechnology. So what are the types already we discussed? Red biotechnology and white biotechnology. Green and blue, particularly lead mostly used for where, in the organism to produce new drugs. And white biotechnology mostly used for where, the fuel chemical substances. Involves the industrial process such as the production of new chemicals or the development of the new fuels for vehicles. This is considered as a white biotechnology. Next one is called as a green biotechnology. Green means already you know a little bit. What is meant by green? Already you know a little about nothing but the agriculture. Okay, so agriculture oriented. Green biotechnology applies to agriculture, and the development is also and involves such process as the development of the pest-resistant grains, not another grains. Agriculture related product, whatever it may be, it is related to which one, genetically. What is that? Development of pest resistant. It is very, very important for the commercial purpose. Suppose when you are planting any one of the plant, commercial oriented, whether it may be a pulses or cereals or any other product in fruits and vegetables also. Suppose if it is not has the pest resistant capacity, what happened? The yield is completely drop out. So the farmers as well as the commercial purpose, it will be completely lost their profit, isn't it? That is very, very important. So the green biotechnology is a very, very important one. There in this technology, what they are using, the development of the pest resistant grains or the accelerated of the evolution of the disease resistant animals. We will see in this category how it will be resistant animals also produced. Okay, that things. Okay, what are the next one is the last and the least. Okay, blue biotechnology. The blue biotechnology rarely mentioned encompasses process in marine and aquatic. Blue means already the sea color is blue, or marine color is blue. Okay, that's why it is mostly used for where marine and aquatic environment. Okay, much as controlling the proliferation of the noxious waterborne organism, noxious, otherwise called as toxious, whatever it may be. Unwanted things, unwanted. Okay, which one is very unwanted things, unwanted microorganism, unwanted animals present in it. Okay, such things are reduced, particularly waterborne organism, aquatic organism. So it will destroy the aquatic atmosphere. So they can completely remove with the help of the technology. Such type of, for example, take an example. So Pseudomonas. Okay, Pseudomonas is one of the special biotechnology introduced microorganism. What is the main uses of this microorganism? They can split the which one? They can split the petrochemical. Petrochemical, sometimes what happen, when the ship is going on, that time the tide is over. That time what they will do, completely pour the which one, crude oil on into the sea. As a result, what happened? The force of the ties will be reduced because here there is a what is that? Friction force is also very slow in nature. As a result, what happened? The ship will be escape from the ties. So after that, what happened? The completely the petrochemical on the surface, it will not remove easily. How can you remove it? So the aquatic animals completely what happened? Suffer. They cannot be able to breathe. The surface water molecule, surface aquatic animals also, they cannot be able to breathe. They cannot be able to reproduce themselves. So that time what that happened? They were spreading the which one? One of the special microorganism is called a Pseudomonas pittica. Pseudomonas pittica, the particular name of the microorganism. They can spread in the form of powder. So as a result, what happened? It acts upon the hydrocarbon petrochemicals. It will be split into simple hydrogen carbon carbon. As a result, what happened? The surface of the bed, sea bed, is completely clean or cured. Okay. So as a result, what happened? The aquatic animals are easily what happened? Survive in the particular area. So this is also one of the blue biotechnological development. Okay. So these are the things. So you have to note it down. This is very, very important. Sometimes they may ask the question in this part. Next, we will pass on to the main heading is genetic engineering. Biotechnology means, what is meant by genetic engineer? Who is engineer? He framed his own style, isn't it? Any one of the construction, okay, is called as what is a civil engineer. Here they told which one, genetic engineer. Who is the genetic engineer? Genetically, we can construct our own gene is called as genetic engineer, isn't it? Okay. What are they? How it will be used? See that? The term genetic engineer refer to a number of new techniques involving the transfer of specific genetic information from one organism to another. So that is called as genetic engineering. That is the name. What is the meaning of that one? We can construct our own gene, our own style. That type of technology is called as genetic engineering. Already we discussed that things. Two strain we may obtain, whether it may be A strain or B strains. Both are having the extra chromosomal body is called as plasmid, and also it has the ability to restriction of some characteristics, isn't it? We can identify the particular characteristics, genes, and also cut with the help of the molecular sister, and then they may be united with the help of the technologies called as, what is that? Hybridization. With the help of the enzyme is called as DNA leakages. After that, what happened? And it will introduce into the specific course. Then the replication is also proceed. Okay. So this is very, very important, my dear students. See, that next one is this take NEET to do not rely on the sexual method. It is not a sexual method. Okay, but instead they involve the genetic manipulation. So the manually we are doing, that is called as manipulation, otherwise called as at a cellular and molecular level. What is the meaning of cellular level? We can construct the cell aspects also. And molecular means, take an example. So what are the molecules? DNA, RNA, or any other protein molecules, enzymes, whatever it may be, hormones. These are all considered molecules. We can change the structure, shape of this one, which one due to the genetic engineering. That is very, very important. Yesterday we saw it, isn't it? The pig. Insulin also similar to that of the human being, isn't it? What are the changes? The physiological changes occurs within the molecules. Then only it will be act as a human insulins. Otherwise, it is as it is in the animal insulin. The bridge is also already in our, but there is a disulfide bridge is there. Okay. And human insulin also, it has a, what is that? The same structure. But some of the fragments we can remove or add. That is the addition or deletion process. That is also genetical abnormalities. Okay. That things is also very, very important in this technique. What are the next one? These are the non-sexual method for the gene transfer. Genetic engineering is now considered as a kind of biotechnology. Okay, it's nothing but a type of biotechnology. Genetic engineering is a part of the biotechnology. Okay, this is very, very important, my dear students. You understand? Okay. Next, the technique of the genetic engineering include, what are the technique they are using? They have been reported. There are three types. One is called as creation of recombinant DNA. Creation of recombinant. Recombinant means different strain united together and forms a single DNA structure. It's called as creation of the recombinant DNA. After that, use of gene cloning. What is the meaning of cloning? We may construct our own DNA, right? Okay. Then how it will be introduced into the, how it will be produced the new one with the help of the cloning methodology, cloning. Okay, use of gene cloning. During the gene cloning, you may obtain any one of the parent cell and isolate the DNA molecule. Again, it will be replaced in the same one. That condition is called as gene cloning. Already I told you. One of the scientists, Ian Wilmot, in 1996, Dolly goat was invented with the help of the uniparental. Okay, by the process is called a gene cloning. We'll see it in the process. Next one is gene transfer. You are isolated a specific gene also isolated by the method is called as already we know about gel electrophoresis or whatever it may be. You isolate it, right? Okay. How it will be transferred into the cell. There is a special method is there. Then only it will be entered. Okay. So that condition is called as, what is that? That process is called as gene transfer. These are the three things is very, very important for genetic engineering. Without these three things, there is no genetic engineering. Okay. So what are they? First one is creation of recombinant DNA technology. DNA means both. Okay. Desired quality. isolated from the different genes and united together, combine. I will get the one single copy of the DNA, both the character present in it. Second one is use of gene cloning. After that, it may be enter into the cloning process, and gene transfer is also. These are the very, very important. And next, we'll pass on to the next heading is genetic modification has. What are the genetic modification? Why we are doing? What are the genetic modification it has? How it will be used for the plant as well as the animals. Okay. See that one by one. It has been reported. How many types? 5 have been given here. One is mad crab more tolerant to abiotic factors. More tolerant. What is the meaning of tolerant? Tolerant is nothing but what is that? Abiotic factor means you can imagine what are the non-living. Outer side is that light, temperature, water molecules, and carbon dioxide, and also oxygen concentration. These are all are considered the headings known as temperature. These are all are considered the headings known as what is that? Abiotic factor. See that one? Cold, draft, salt, heat. These are all are called as what is that? Abiotic factor. So when we are producing this type of plants, what happens? They may fight against these factors. Such type of organisms, such as the plant or animals, we can produce. That is called as newly formed organism, newly formed plants. That is called as genetics. What is that? Modification has. We can do only the genetically modification only. How it will be? There is a technical that we discussed. Second one is reduce reliance on chemical pesticide. Okay. We have to reduce the pesticide resistant crops also. Some of the pesticides, it will be completely destroy the yield. Okay. In such cases, we have no idea. In previous cases, there is no idea for this pesticides. We are using some chemical substances, natural substances. Take an example, neem cake and also tobacco. These are all are mixed with the water and also spray it. Nowadays, there are so many chemicals, so many pesticides are introduced. Okay. So this is also one of the another things. Third one is help to reduce the post-harvest losses. Post-harvest previously. We cannot able to harvest the things. Okay. Some of them are what happened now. Post-harvest, suppose if you want to cultivate the pulses or cereals, normal growth is already in our, but there are 3 months or 4 months, isn't it? Isn't it? Suppose if you want to harvest within 40 days, how will you? Nowadays, there are so many rice varieties have been identified. 40 days, 30 days, 60 days. These are all are considered the headings known as post-harvest losses. And then fourth one is increase the efficiency of mineral usage by plant, because all the minerals is not utilized by the plant. Okay, when you are giving the substances, okay, when you are introducing the fertilizers, otherwise, suppose the soil already naturally present in it, isn't it? That components, how it will be absorbed? They will not absorb. Increase the efficiency. How it will be increased? By this process only, genetically modification only we can do it, the things. To increase the efficiency of the mineral usage by plant. This prevents easily exhaust of fertility of the soils. This is also very, very, what is that? Exhaust of fertility of the soil. This is very, 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 what is that? Danger also. Okay. And go to the last one. Enhance the nutritional value of food also. Suppose take an examples. Nowadays, there are so many variety of rice is also introduced newly. They are all genetically modified organisms. Take an examples of vitamin A enriched rice also. Carotenoid containing rice also. Nowadays, they were introduced. Take an example, pumpkin. Normal pumpkin, it may be in 1.5 kg. Now they produce for 20 kg, 15 kg, 10 kg, like that. Even sugar cane is also, there is a particular height only, maximum 6 feet, isn't it? Nowadays, 20 feet. And take an example, Chinese peoples, even seedless fruits, okay, grapes and whatever it may be, they are all are newly invented. Take an example, what is a brinjal, chilies, seedless chilies, and the size also very big. They are all are considered in commercial value. They are all are considered the commercial value. These things also done by the genetically modification has. What are they? See that one. Mad craft more tolerant to abiotic stresses, cold, drought, salt, and heat. Second one is reduce the reliance on chemical pesticides. Second things. Third one is helped to reduce the post-harvest losses. Post-harvest losses. And increase the efficiency of mineral usage by plant. Suppose some of the plant they will not utilize completely, isn't it? We have to increase the usage of the plant. And also they enhance the nutritional value of food also. Not only that, vitamin A enriched rice, carotenoid rice, and so many things is there. Okay, take an example of ginger, sizes also. Pumpkin, same thing, size. Okay, suppose take an examples of fruits. In sugar cane, the sugar content of you have to rise, it will be rice. Okay, so likewise, all are it has a own specific, what is that? Nutritional value. Which one is a special characteristics? Sugar cane, it has a sugar only, the content of the sugar, isn't it? It may increase due to the enhanced the nutritional value. It is also one more is that. Vitamin enriched A, vitamin A containing, that is also mostly found in where, maximum orange containing food only. But in this category, vitamin A is also present in the rice. We can take rice, you will get vitamin A. Okay. So likewise, it is also made by which one, genetic modifications. Next, we will pass on to the next one is very, very important application of the genetic engineering. What are the importance? Okay. What are the main uses of the genetic engineering? What are the applications? The cloning genes to cloning organisms, including transgenic microbes. This is very, very important one. First point, cloning genes to the cloning organisms, including the transgenic microbes. Not only that, not only the microorganism. And we can do it in the animals also. And second one is called a genetically modified food product. Already told you, cabbage, chili, okay, and seedless fruits, grapes, sapota, mango, pineapples. Okay, pineapple already you know, but its size will be depends on the size. Size can be increase due to this one genetically modified organisms, cabbage, brinjal, okay, pumpkins. Those are having the commercial value. Size is very, very important. Some of them are nutritional aspect. The content, inner content is very, very important. Just such as a carbohydrate. Okay, likewise. And third one is a human gene product, pharmacologically and therapeutically useful product. Genetically, already we discussed which one is the resistant gene. Is some of them are resistant, isn't it? Some of them they do not have the resistant power. So that things also done by which one, the applications of genetic engineering. We can do it. Gene replacement and gene augmentations therapy. Gene replacement, where it will be do, suppose the person suffering from any one of the diseases, we can easily identify it before birth, during the embryo stage. We can replace the gene also. As a result, the genetical disorder, that means the one generation to another generation, it will not continue. Some of the diseases also present in the previous generations. We can identify and remove that particular gene during the embryo stage. What happened to the progeny, it will not contain that type of gene. So the disease or any other gene, it will not continue to the next generation. It will completely stop in that generation itself. So such type of application mostly in the genetic engineering. And then DNA fingerprinting has an immense application for forensic investigations. Already we discussed these things also. What is by fingerprinting? Forensic means you know about it. Who did it? We do not know. Suspected persons, sometimes it may be used as mostly in the uniform service they were used. Any other bomb blast or culprit. Okay. Murder, robbery, whatever it may be. They can use it. The DNA fingerprinting. Another one is the disappearing of the human genome has revealed the blueprint of our life itself. This is another one. Okay. And then third of, and then rise the disease resistant high yielding variety of crops. Already we discussed abiotic factors. What are the abiotic factors? Climate. Some of the plant they can tolerate the high climate. Suppose take an example, tea, only cultivated in the slope region only. Any one of the hills, there is a slope region. They will not need much amount of water in their soil. Okay, but it will not grow in the tropical region because the temperature is very high at the same time. What happened now? Whether it may be a water content is also. That time what happened, they will not get the proper yield. Okay, so they can survive only in the particular area. Only the climate is very, very essential. It also cotton is also cotton, rice, and bamboo, and sugar cane is also other vegetables, pulses, cereals, and so on. So many things is there. They are all, or what is that? We can do it in this process. Introduce a biocide in agriculture. Already we discussed this. Some of the biocides. Biocides means one microorganism kill the another one. In the agricultural aspects, we'll see BT cotton. Already we discussed the things also in the DNA fingerprinting, the technology already you know about. Okay, so these are all comes under the getting known as what is that? DNA genetic engineering application point of view. And next one is preserve germ blossom of the plant and animal and microbes. Suppose some of the germs is very, very essential for the biotechnology or genetical aspects. Such type of germs. Take an example, COVID also, COVID-19, isn't it? Nowadays, the PF7 is also there. These are all our germs. They were used for the bio war. Already we know. Okay. That germs also used sometimes in the plant in the beneficial aspect. Such type of animals, that such type of plants, such type of microorganisms may be stored in the lab. Okay. In beneficial aspect. Suppose if it is used as a harmful effect, the bio war, just like as a COVID in China. Okay, they were used. Okay. The next one is produce pharmaceutical product. Already we discussed it. Some of the pharmaceutical product also done by the genetic engineering. Produce biofuels. Biofuels means microorganisms used as a fuels. Okay, production. Okay, so already we discussed sphagnum is the one of the fens used for the production of biofuels. Okay, media. And then use of various microorganisms in food making and preservations. This is also very, very important. Take an examples. Microorganism. Which microorganism used as a preservative? Normally, any one of the food product commercially prepared, they were used some chemical substances for the preservation. Take an example, sodium todicile. Okay. And sodium citrate. The other chemical substances also there. Each and every product day by day we are using. In this category, okay, there is a preservative is there. Some of the microorganisms act as a preservative. Okay, so we need that preservatives. So it act as a preservative means the content is also very, very important. They can be stored in the lab. Okay, and then employ a microorganism in the extraction of minerals from ore, minimize pollution hazard. This is also a commercial oriented. Some of the microorganism used as a which one extraction of mineral from the ore. Okay, if you want to isolate the minerals particularly ions, copper, sulfur, phosphate, nitrogen. Nitrogen is not there. It's any other. Okay. Water content, mineral ores. What are the minerals we can absorb or extracted from the other part? That time they were used the microorganism. What happened? The microorganism act as used for the extraction of minerals from the ore only. So such type cases, what happened? That the applications also important. Which one? Genetic engineering. Next, we will pass on to the next one is very, very important, my dear students. This is a very, very important one. So gene cloning. So this is very, very, very important. This is very, very important. What is by gene cloning? The term gene cloning can be defined as the isolation and amplification of an individual gene sequence by insertion of that sequence into a bacterium or other cell where it can be replicated. That is very, very important. This is the point. What is that? Suppose we are isolating the microorganism in a particular, what is that? The term gene cloning can be defined as the isolation and amplification of an individual gene's sequence by insertion of that sequence into a bacterium or other cell where it can be replicated. Then only the gene cloning will be success. Otherwise, what happens? The gene cloning will be not success. Okay. So the gene cloning is a very, very important things. Generally, you can create the new combination, recombinant animals or plants, whatever it may be. Okay. The cloning experiments involves, what are the cloning experiments? How it will be involved? What are the process which occurs? Gene cloning. Listen, the first one is the isolation of the gene of the interest. Already we discussed that things also. Which gene is important for us? That gene must be isolated from the other organisms. See that a fragment of DNA, otherwise called a gene insert, to be cloned is incorporated into a small replicating, usually circular in manner, that is called as plasmid. The DNA molecule is called as vector. So that is nothing but the vector is what is that? A small fragment DNA molecule, which one we are going to insert to the another one. Such DNA molecule is called as vector. It is usually a which nature, circular nature. Okay. The vector with the incorporated gene is called as recombinant DNA molecules. Which molecules we are going to incorporate? Such gene is called as what is that? So such gene is called as what is that? See this one? You can easily identify. Generally, it will be better. So this is called as what is that? Vector gene. Okay. So this is very, very important idea. So these are all are concepts that is known as cloning experiments. Cloning experiment involve what are they now? First one is you have to isolate the gene interest. Okay. Isolate the gene interest. Copy here. So it will be better for your understanding. Then what happened? The recombinant vector is introduced into the host cell by transformation, by the process called as transformation, gene transformation from the one organism to another organisms, selected one. Then cells that have acquired recombinant DNA molecules are selected. Recombinant DNA molecule is multiplied within the host cell to produce a number of identical copy of the cloned genes. This one is already you know all about. So for example, see that one? Already we discussed. So take an example. This one is considered a strain. So this one is considered as a B strain. Okay, or bacteria, A bacteria, B bacteria. So inside there is a but there is a molecule is there. So this is considered as a DNA molecule. Yesterday we saw it, this point also already discussed. This two molecule. This is nothing but the circular. What is the meaning of circular? Circular DNA molecule. Both. The circular DNA molecule is called as, what is that? Simply we may call as, what is that? Plasmid plus plasmid. Okay. This is a circular plasmid. Both are same. Here already we discussed which one is the desired quality. So some of them are, take an example, vitamin A rich component gene. The particular portion. This portion is responsible for the vitamin A. Okay. So vitamin A gene. So in this portion, the same things is also, you have to isolate it from this portion. Okay. Here we have to isolate these things also. Again, it has a specific methodology for the, which one the separation of molecules. Okay. Already yesterday we saw it, these things also. The particular, again, the same process. The particular gene is removed. This particular area will be removed. Okay. So as a result, what happened now? It's look like what off only you will get it, isn't it? So this portion will get it. Okay. This portion completely removed. Okay. Likewise, here also same. Here also, what happened now? The structure is look like this one. Okay. Structure is look like this one. The portion is completely removed. This is responsible for the, okay, the vitamin A rich DNA molecules. Now what happened, this DNA vitamin rich A components gene is also gene transformation takes place where in the B to A. So like this. Okay, this is the B genes. Okay, just imagine. Now, after the recombination completed, this can be introduced into the host. This can be introduced here. What happened now? This is the A gene. This one is called as, what is the B gene? Isn't it? B responsible. This gene is here. So both are introduced into the new vector. That vector is mostly in the bacteriophage. Take an example. This is called as vector. This is the vector. Okay. This is a step, say, first step. Second, we are removing, and we'll get the structure like this. Third one. Here also same. This is the first. And second, identify. Third one is, we made a cut and also isolated with the help of the molecular scissor. Then united. Okay. After that, it will be introduced into the host cell. That host must be which one? See this one. The structure will be like this. After that, it has a method, specific method. There is a micro pipette, and sometimes they were used the gun also, electric gun also is that they were used for target tissues. See that? So now introduce inside the host. Okay, so this is the fourth part. Fifth process. Then now what happened, the inside the host, this is the host, maximum they were used the E. coli as the host. E. coli, host. Okay, maximum they were used the host E. coli. E. coli. E. coli, bacteria used as a host. So here what happened? Both are same. These are also bacillus bacteria. This one is called as E. coli bacteria. When it inserted these two combined recombinant DNA molecule within the host, what happened? It undergoes the replication process. You will obtain what? Both the combination, A and B genes present is inside it. Again, it be introduced into the where directly into the plant. Directly into the plant. So now what happened? Now the plant yield, it contains, what is that? The plant yield is contain vitamin A. So that is the also new variety. So this is the important. Okay, are you clear now? So this is very, very important, my dear students. This is very, very important, have been reported in your part. See these things. So you will get a number of identical copy of the cloned gene. Which one is cloned inside the host will obtain the number of copies. How many copies we need? That copy completely produced inside the host only. That host is, see here, the host cell is very, very important. Without host, what happened now? We will not, it will not complete the, what is that process. This is very, very important. Okay. So this is the important thing. After that, see that one. See. So what are the process which occurs? First of all, gene cloning is a technique. How it will be happen? See, these are the steps. Is very, very important. There are 3 basic steps involved in the DNA cloning technology. First one is isolation of the DNA extraction is the first process. First, you should isolate from the molecules from the different strains. Different strain means already I saw it, isn't it? Which one is different strain? So this one taken. These 2 are different strain, isn't it? These different strains, we have to extract the DNA molecule from outside within the cell to come out from the outside. So we have to rupture the cell wall, isn't it? Then only it comes out. Likewise here also, rupture the cell wall. After getting the DNA plasmids in the vector, we can isolate, identify which area is responsible or which gene is responsible for the characteristics, which characteristics if you want to clone. Identify the gene, location, particular location in chromosome, location or loci. Then we have to use the restriction enzyme for cutting process, that is act as a molecular scissor. After that molecular scissor means what happened, you will get it the fragments just like this one. So you will get the fragments. Remove this part, you will get the fragments. Here also, same process also do the next one. B strain also. Okay. From this part, the B strains, it has a vitamin A containing gene. After the identification, it may be transfer the gene to the B2A. After the completion, when we are transferring the gene molecule from the B strain to A strain with the help of the ligase enzyme, the recombinant is completed. No. After the completion, he introduced into the host cell for the replication process. After the replication process only, what happened, the DNA genetic or gene cloning is completed. This process called as, after this, he introduced into the host cell, this process called as gene cloning. Taken, isolated, and also recombination done. After that, it's also cloning process finished. Okay. After that, what happened? The number of copies we will obtain in both the genes present inside. It is also introduced into the in the respective plants or animals, whatever it may be. It produces the vitamin A containing component inside the plant. That is the final step involved in the methodology of gene cloning or genetic engineering. Okay, so this is the very one. See here, DNA extractions. The isolation of genetic material DNA from cell or bacteria, viral, plant or animal. That thing's also already told you. Any one of the plant or animal sources, we have to isolate the genetic material. Genetic material already know, DNA is genetic material, RNA is also genetic material, but it is not complete, isn't it? Already we discussed. So what are the 3 steps that were involved for the DNA extraction? First of all, rupturing of cell membrane to release the cellular component on the DNA. This is already we discussed, isn't it? First of all, this is a important point. Already we discussed. Okay. So this is very, very important. Rupturing the cell membrane, then only the inner content will be comes out. Next one is, what is that, the separation of nucleic acid. Separation of nucleic acid from the cellular component. After that, it undergoes the purification process. After the purification only, what happened now? He will undergo the cloning process. Then only the yield will be purified in nature. Suppose if it is not properly done, mean you will get the improper cloning. So the yield is not good. That is very, very important. So the DNA extraction, what are the three steps, three basic steps they were used, First of all, rupturing of the cell membrane to release the cellular component and the DNA. Separation of nucleic acid from other cellular component. And the last one is purification of nucleic acid. After that, only the process will be undergoes. For tissue culture cells and blood samples, incubation of buffer containing detergent such as sodium toadyl sulfate. This is very, very important medicine. Suppose after the collection of the samples, you have to introduce the one of the buffer system, the buffer containing detergent such as sodium toadyl sulfate. Don't forget, my dear students, SDS is very, very important. Mostly used for the genetic engineering, particularly in the laboratory aspect, storage purpose after the sample collection. Is usually sufficient to disturb the cell membrane and release high molecular weight DNA. What happens, the detergent molecule, it can bind with the cell membrane of the plant or animal cell. What happens? Automatically, the cells that lose their ability, cell ability, the flexibility. So as a result, what happens? It ruptures easily. So inner content easily comes out, particularly to release the high molecule compared to the cell. Any one of the cell, you can take an example, any one of the cell membrane cell, the nucleus, the high molecular weight compared to the other components such as endoplasmic reticulum, ribosomes, mitochondria, and Golgi apparatus, dictyosomes, okay, vacuole, and whatever it may be. The DNA is a high molecular weight present inside the cell. One, it can be easily comes out due to the one of the chemical substances. What is that? Sodium toadyl sulfate. This is very, very important, my dear students. Don't forget. After that, cells are lyzed open to release the DNA along with other macromolecules such as RNA, protein, polysaccharide, and also lipids. Not only the DNA molecule. What are the other biomolecules present in it? They are all are considered it is macromolecules, isn't it? These are all comes out, lyzed open to release the DNA along with the other macromolecules such as RNA, protein, polysaccharide, and also lipids. Then RNA can be removed by treatment with the ribonuclease. Already know very about. Other molecule can be removed by appropriate treatment. Each one it has a specific treatment. Protein separation and also RNA separation with the help of the ribonuclease. DNA also electrophoresis, and protein also, that is also one of the chromatographic technique. So many things is there, okay? They may be isolated, different according to their molecular weight. After the removal of protein, DNA molecules will be present at the interface. Complete removal of the molecules. Completely removal of the molecules. Which one is contained? DNA is a high molecular weight present inside the interface, inside. Then precipitation of the DNA is brought about by the using cold chilled ethanol. This is very, very important. How will you identify, okay, the DNA molecule from the component? With the help of the chilled ethanol. This is very, very important. Cold or chilled ethanol. Ethanol is very, very important for the precipitation of the DNA molecule from the sample. After that, this can be seen as a collection of fine thread in the suspension. After precipitation, it is centrifuged. DNA is insoluble in alcohol. Therefore, it comes out of the solution immediately. Okay. So this is very, very important. How the DNA molecules also isolated Okay. So that things is we will finish it in today. This lesson is finished in this level only. So today, what are the things we will discuss one by one? We will discuss here. What are the types of, what is that? Biotechnology. First one is red and white and green and blue. Red biotechnology already we discussed is mostly used for the production of new drugs. White biotechnology mostly used for the, which one you know? The new chemical and also new fuels for the vehicles. That's why it is called as a gray biotechnology. What is by green biotechnology? Mostly used for the agriculture purpose, particularly the development of the pest-resistant grains. The blue biotechnology, marine and aquatic, mostly used for the proliferation of noxious waterborne organisms. And the next one is genetic engineering. What is my genetic engineering? The term genetic engineering refers to a number of new techniques involve the transfer of the specific gene information from one organism to another organism. Which one is the desired character we need valuable in public or populations. The techniques do not rely on the sexual method, but instead they involve the genetic manipulation at the cellular and molecular level. So molecular level is already we discussed DNA as well as RNA molecules. But these are the non-sexual method. Once again, I repeat for you that this is very, very important point of view. Non-sexual method, which one of the following is a non-sexual method? They may give the absence, genetic engineering, other component also. So you have to identify the genetic engineering, which one is the non-sexual method. It is also one of the, which one you know, biotechnology, branch of biotechnology, kinds of biotechnology. The next one is already we should know very well. The technique of the genetic engineering included 3 steps is very, very important. What are they? First one is a creation of the recombinant DNA. Different strain is that different strains are united together and form as a single gene that is called as recombinant DNA. After that, the gene is used for cloning purpose. During cloning process, what are the transfer material is also used, vector and also another one, and also the cost is also. So these are the 3 steps is very, very important idea. So next one is the genetic modification has, what are the modification, what are the uses of this one, and also their application, how it will be used for the public or human being. Genetic modification has the made crop more tolerant to abiotic factors, abiotic stresses, cold, drought, salt, heat, etc. Reduce reliance on the chemical pesticide, pest-resistant crops also. This is also important things. Help to reduce the post-harvest losses, increase the efficiency of mineral usage by plant. This prevent easily exhausting the fertility of the soil. And the last one is the enhance the nutritional value of food. Example, vitamin A enriched rice is also produced by this method. Okay. And application already we discussed the cloning genes or to cloning organism included transgenic microbes, genetically modified food products, human gene produced pharmacologically and therapeutically useful products, gene replacement or gene augmentation therapy is also very, very important for the fetus. Okay. And the DNA fingerprinting has immense application for the forensic investigation. Disappearing of the human genome has revealed the blueprint of our life itself, raised disease resistant, high yielding variety of crops, and introduced biocide in agriculture. Okay. And then preserve a germplasm of plant, animal, and microbes in the lab, produce pharmaceutical product and produce biofuels also in commercial aspects, with the help of the biotechnology or genetic engineering, use of various microorganisms in food making and preservation process also, employ microorganisms in the extraction of the minerals from the ore. This is a very, very important thing. Minimize pollution hazards. Okay. These are all the main important application of the genetic engineering. Next method already in our the gene cloning. Okay. The term gene cloning can be defined as the isolation and the amplification of an individual gene sequence by insertion of the sequence into the bacterium or the cell where it can be replicate. That process is called gene cloning. The gene cloning experiment involved the following steps. Particularly, first one is called as isolation of gene interest. Which gene is we want to isolate, we want to reproduce it by the technology. Okay, by the usage of this technology, the gene is called as, what is that? Isolate. How it will be isolated? See that one? A fragment of DNA gene inserted to be incorporated into a small replicating DNA molecule is called as vector. The vector with the incorporated gene is called as the recombinant DNA molecules. After the isolations, what happened? You have to introduce into the both gene into the recombinant technology and also introduce into the host. Okay. The recombinant vector is introduced into the host cell by transformations after the isolations. The cell that have the acute recombinant DNA molecule are selected. That is very, very important. Very, very important point, this one. Okay. Unwantedly, we will not use it, this technology. Wanted character, acute recombinant DNA molecule are selected one. And recombination DNA molecules is multiply within the host cell to produce a number of identical copies of the cloned gene also. Very, very important. After that, DNA extraction. Already in overlay, there are 3 steps are involved in this process. Rupturing of cell membrane to release the cellular component and DNA. Separation of the nucleic acid from other cellular component. Purification of the nucleic acid. Okay, these are the very important steps. And which component after the cells, isolated cells, we have to use it. Sodium sulfate, SDSG. It is used for the, what is the storage purpose. Then only what happened, this chemical substance otherwise called as detergent. It is also one of the buffer containing detergent. It can be easily rupture the cell wall and the inner molecules, cell bound organism comes out very easily. Okay, not only the DNA molecule and other component, other high molecular weights such as which one, RNA and protein and polysaccharide and also lipids also it comes out easily with the help of these components. So it is used as a easily isolate the internal components. That is very, very important, my dear student. Okay, so then what happened? Break the cell and also with the help of the enzymes. Okay, that things we will see in the next classes, my dear. Thank you so much for the participation. We'll see the remaining things on the next class. Thank you so much. Bye. Take care. Good night. Thank you, ma'am.
Thank you, sir.